Okay. Uh, okay, so we'll continue with our discussion. Okay, but before that, I want to discuss regarding your regarding the practice one. Okay. Okay. Saya share. So okay. Okay. Okay, so this is just an example from one of you. Okay, regarding the practice one. Okay, so this is the data. Okay, so if we were to look at the data, okay, so this is a person's name, person's height, okay, and then this is the cumulative. So it is the same because it is just one person. And then you have the average height, okay, it is still the same because it is just one person. And the difference, okay, so this is the difference between this height, okay, and the population mean, which is this one. Okay. Okay. How do I enlarge this? Okay, I hope all of you can see it clearly. Okay, and then for this one, so you can see that the difference is 2.2 cm. So meaning that if you were to take at random one individual, then it is not going to be exactly the same, okay, with the population mean. Okay, so the mean that we get from just one data is different from the actual population mean that represents the population. Okay, so that is why we we need to when we want to estimate, okay, we want to be as close as possible to the population mean because we don't really know what the population mean is. So what we can do is to estimate. Okay, it's just to estimate. Okay, so then we take another one, okay, randomly. So the Hanisa's height is 151 cm. So the cumulative meaning 151 plus 160 equals to 311. And then so the average height, okay, 311 divided by 2 will be 155.5. Okay, so the difference is 2.3. Okay, about the same. Okay, and then Kamarina, when we add Kamarina's data, then the difference becomes 3.1. Okay, so we can look at the graph. Okay, so this is the number of person. Okay, so initially, if we were to take just one, then the difference with the population mean is 2.2. It increases, and then when we add Kamarina, so the difference shoot up. Okay, so but then it starts to go down. Okay, so this is done by random. Okay, randomly. So meaning that, okay, if we were to look at the overall trend, okay, the more the number of person, then the difference between the estimated mean, which is this one, okay, the average height. This is the estimated mean of the population, okay, and the actual population mean will decrease. Okay, so the more the sample that we take from the population, the better the estimation of the population mean, the closer that we will get to the actual population mean. Okay, that is in general, that is correct. Okay, but there will always be something, there will always be exceptions. Okay, for example, like this one. Okay, Okay, Camarina is in. Okay, so when we add Camarina over here, then it shoot up. Okay, even though previously it is just one sample, and then it is two samples. But when we add the third sample, then the difference increases. Okay, so that is why statistics is based on probability. It is not an exact, okay, determination or measurement. Okay, it is based on probability. Even though we say that, and in most cases it is true, okay, even though we say that when we increase the number of samples, okay, then we will get a better estimation, a closer estimation to the actual population mean. Okay, but when we look at this, then actually it is worse off. We are worse off because the difference is higher. Okay, that happens. Okay, there is no guarantee that when we increase the number of samples, then our estimated population mean will be closer or exactly the same as the actual population mean. But okay, the possibility of that happening is lesser compared to us, okay, getting a better estimation to the population mean when we increase our number of samples. Okay, so this is the nature of statistics that we have discussed previously. It is not the same as mathematics. Mathematics, you have exact answers. Okay, 2 plus 2 is definitely going to be 4. It cannot be 4.0001 even. It has to be exactly 4. But when it comes to statistics, there is always a possibility that exceptions will occur. Okay, but we are still better off because most by taking more than one sample, because in most cases it will improve our estimation of our mean closer to the actual population mean. Okay. Okay, so this is just to demonstrate to you the effect of replication and also the importance of doing replication. That is why when, especially in food science, where the degree of heterogeneity, this is the showing you the effect of heterogeneity. Okay, so you can see that this is the heterogeneity of the height of the population. Okay, so usually when we are dealing with biological samples, then we have a higher degree of heterogeneity. That is why one data doesn't mean anything. Okay, one data doesn't mean anything. Okay, previously we are okay with 2 replications. Okay, but now the minimum is 3 replications. Okay, so but even though usually we do 3 replications, okay, but it is not a must. Okay, 3 is the minimum, but you can go more than 3. Okay, more than 3. When the degree of heterogeneity is higher. Okay, when you get when you are dealing with samples with a higher degree of heterogeneity, then you need to take more samples. Okay, for example, let me change this one. Okay. 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 Okay, for example, you can remember how are we able to get a better estimation of the population mean? Okay, so basically it is because of the normal distribution. Okay, because of the normal distribution. Okay, so this is the actual population mean. Okay, so this is the actual population mean, and then we take samples, okay, randomly. Okay, but because the majority of the of the population resides near the population mean. Okay, then the probability of taking a sample which is near the population mean is higher compared to the probability of taking a sample further from the population mean. Okay, so it's just a matter of numbers. Okay, because like for example in this class, if I were to take one student, okay, the probability of selecting a female student is much more higher compared to a male student, because the number of female students are more. Okay, so that's why if you were to look at this normal distribution, that is why it is like this bell shaped. Okay, compared to, like for example, if you have like this, okay, same, okay, but it is like that. Okay, this means, okay, so this is number of, or the frequency, or the number of individuals, anything. Okay, similar to this. Okay, so with this one, the nature of this population is that the majority resides around here. That's why it is bell-shaped. Okay, because here the number is more, the number of individuals. This is lesser, this is lesser, this is the least. Okay, this is the least number of individuals. So the nature of the population is that the majority of the population is around the actual population mean. That's why it comes out like this bell-shaped. Compared to this one, okay, in this population, okay, the number of person over here, okay, for example, just take we'll take the height. Okay, this is for height. Okay, so this is also for height. Okay, it in this population, it means that all the individuals have the same height. Okay, okay, so sorry, not the same height, but the same frequency. Okay, for example, okay, if the height is 100 cm over here, so the number is, for example, 10. Okay, if it is 50 cm, it is also 10. So at any height, okay, has the same number of population. Okay, has the same number of population. Okay, so in this case, right here, okay, it doesn't matter if you were to take how many samples. Okay, you will still not get a good estimation of the population mean. Okay, you will still not get a good estimation of the population mean, because the probability of taking an individual with this height over here is the same as over here. It's the same as over here. So it doesn't apply. So that is why usually among the assumptions, okay, or the precondition of statistical tests is that the population follows a normal distribution. Okay, follows a normal distribution. Okay, so again we compare between two. This one is like this. Okay, I'll just put it in the same page. Okay, this one is like this. This one is like this. Both have the same average population mean. Okay, so if you were to compare these two, okay, if you want to take, if for example this one, you take 3 replications. For this one, more or less replications. You have to take more than 3 reps, because the heterogeneity is larger. This one, higher degree of heterogeneity. Okay, so that's why this is higher compared to this one. That's why this is higher compared to this one, because this is more homogeneous. Okay. For example, if it is like this, okay, so it is very low, and then it shoots up there, like the peak in a chromatogram. So this is very homogeneous. Very homogeneous. Okay. Okay. So usually that is why in certain fields they do not do replications, or they do not do statistics, ANOVA, etc. They rely only on the mean, because they are dealing with very homogeneous samples. Okay. Okay, so that is why it is important to do replications. Okay, it is important to do replications. And what is replication? Okay, another aspect. Okay, what is replication? Replication means that, okay, remember, replication we do is to get a better estimation of the population. Okay, if of the population mean. If, for example, we are we want to determine objective to determine the effect of temperature, okay, on color, for example. Okay, so the factor, okay, what effect are we looking at? The effect of temperature. So temperature is the factor. Okay, and in order to determine the effect of temperature, we cannot use only one temperature, right? If the temperature is constant, then we will not be able to determine the effects. Okay, so we have to vary this. So the level, okay, for example, 60, 70, 80 degrees C. Okay, so what are the treatments? The treatments are the temperature, 70 degrees C, 80 degrees C, right? Okay, what are the responses? Responses are our measurement. Okay, pemala yang direkodkan ke apa, something like that. Okay, responses, for example, you want to determine the color. So we look at the, for example, okay, the L asterisk or L star under the CIE color system. Okay. Okay, so replication, reps, you say that it is 3. Okay, so how do we do 3 reps? Okay, first, okay, for example, like in banana. Okay, temperature of blanching, for example, penceluran of blanching. Okay, so first we take bananas, and then, okay, we will blanch. This is a shortcut, okay? Of course, there are several other steps, but just a shortcut to make the story short. Okay, you have bananas, and then you blanch them, okay? And then after that, okay, blanching at 3 different temperature, right? Okay, and then analyze, okay, using a color meter based on L. Okay, okay, so when you go into the lab, okay, so you have your bananas, okay, you have to make sure that everything is as constant as possible. Make everything constant, okay, for example, weight, okay, of the sample, okay, degree of maturity, okay, of course, the variety, barangan or nipa or lemak manis or emas or anything, okay, everything has to be constant, okay, and then you prepare them, okay, and then you do blanching, for 60, and then you randomize them. Okay, so you need to randomize this. Don't do 60 first, 70 second, and 83rd. Okay, you have to randomize them so that we can minimize any effect of confounding factors. Okay, so for example, like we do this one first, this one second, and this one third, for example. Okay, so when you do 70, then you get your sample. Okay, and then you will analyze. And then this one also you will analyze. This one also you will analyze. This is just one replication. Okay, one replication. You only need to get one data from here, one data from here, and one data from here. That is your first replication. Okay, for your second replication, you need to do all over again. Don't use these bananas. Buy another batch of bananas. Okay, and then do again blanching, and then do again the randomization, and then you get your second rep. Okay, that is replication. Okay, remember, okay, replication means that we are going to take different individuals, okay, from the population. Okay, if you were to take this and then measure it 3 times, okay, L1, L2, L3. You do this only one. You measure it 3 times. You will get 3 numbers, 3 data. But that is not replication because, okay, this is called subsampling. Okay, remember like this one. Okay, replication means that we are going to take 3 individuals from here. Okay, 3 individuals. In this case, okay, we only take 1. Okay, from the same batch of bananas. Okay, only 1, and then we apply the treatment only once. Okay, and then you only measure the measurement. Only the measurement is 3 times. Okay, so this average, when we average this out, this is a representation of this one, which is a representation of this one. It is not a 3 only, and this is only one representation of the population. Okay, it is similar to like this. If you want, in this case, if you want to take the average height, okay, average height, instead of taking 3 individuals, A, B, and C, you only take 1, okay, and then you measure the height 3 times. Okay, so that is subsampling. That is not replication. Okay, replication means that you have to do this, okay, start from here 3 times. Okay, so then you will get a data which is representative of the population of bananas. Okay, if not, you will only get a representative of that particular batch of banana. Okay, not the population of bananas. Okay, okay, so that is about replication. Okay, any questions?
Kata apa, you dah follow lah, you dah finding dulu.
Nak tanya, contohnya kalau kita beli bananas tu kan, kita beli 3 terus dari tempat yang sama, tapi kita buat berbeza lah, kira macam contohnya 3 sikat kita beli. Yang tu boleh dikira replication tak? Atau better kita beli 3 bananas daripada 3 lokasi berbeza, ataupun 3 bananas kita beli pada hari yang lain?
Okay. Dia bergantung kepada kita punya objektif. Okay, kalau objektif itu adalah, for example, kita punya objektif adalah untuk kita tentukan to determine, okay, the effect of temperature, okay, on bananas, for example, from, okay, for example, it is from a particular time, from a particular time period. Okay, then you can buy 3 sikat lah. Okay, sorry. Then you can buy 3 groups of banana. Then you have, okay, group 1, okay, and then you have group 2, and then you have group 3. And then you do this 60, 70, 80. Okay, so this will be a representation of the banana from that particular time period. But in the majority of cases, okay, what we want to do is we want to get a representation of the bananas, regardless of the time period, right? Because usually our research will endure a certain number of years. Okay, so and we know bananas or any plant material, it will be affected by the rain, okay, by the sun. Okay, so I would prefer, okay, to take different batches throughout the year. For example, like this is January, you take one batch of bananas, okay, and then you do the treatment. So this will be your rep 1. And then afterwards, okay, maybe in March or in April or in May, okay, and then you take another batch of bananas, you do again, okay, this will be your rep 2. And then in October, for example, you take bananas, okay, 3, this will be your rep 3. Like that. So whatever that you get, then you take the average. So this number represents the banana. This number represents this population of bananas. So whatever, regardless of the weather, whether it rains or it suns throughout the year, because you have captured that, all the confounding factors, you have captured that, okay, by taking samples throughout the year. Okay, so to me, this is a more representative. Of course, the bananas, okay, you can take from one locality, same locality, or same location. Okay, because usually we do not want it to be if you want it to represent the whole Malaysia, then you have to take in many locations. But in most cases, okay, because we are not doing a survey, okay, we want to know what happens to this particular banana, then location is not in our interest. Okay, location is not in our interest. So it is enough, okay, so it is enough to do from one location. But if your objective is to get, for example, the color of bananas in Malaysia, then you have to take from more locations because you want it to represent Malaysia. But if you are only interested in the bananas, okay, then it is okay enough to take from the same location. But I would prefer that it is taken samples throughout the year to represent, okay, the changes in, if any, if there are any, okay, in the bananas throughout the year. Okay. Any other questions? Okay. Okay, so if there are no other questions, okay, so let's get back to our lecture. Okay. Okay, so we have discussed, okay, previously we have started to discuss regarding the design of experiment. Okay, so just a recap. Okay, so we discussed about why we need to use statistics, especially in food science, basically because we are dealing with samples with a higher degree of heterogeneity. So we need statistics. Okay, so we have also discussed about the function of statistics. Okay, Okay, the function of statistics, and about the nature of statistics based on probability, based on assumptions, and we have discussed that and have been demonstrated by your in your practice one just now. Okay, the basis of experimental statistics. Okay, population follows normal distribution. Okay, and we also have discussed it just now. Very important. Okay, and we have also discussed about the components of statistics, hypothesis, randomization. Okay, replication just now. Okay, mean and standard deviation. Okay, so let's go to our next topic, and that is design of experiment. Okay, actually we have touched a little bit on this when we discussed about your practice one and the example that I have given. Okay, so why do we do experiments? Okay, so many reasons. Okay, basically we want to get the truth. Okay, and that is why it is termed as scientific. Okay, scientific. Okay, it is a scientific experiment and a non-scientific experiment. Okay, these are two different things. Okay, for example, like in our field, that is food science. Okay, what you are doing, for example, okay, if you are taking an ingredient or a compound, put it inside food, and then you determine what are the effects. Okay, so a common practice in food science, because you want to look at the effects of certain things, etc. Okay, but okay, we are food scientists, but there are also chefs in the restaurant, in the hotels. They also do the same thing. They put things inside and then they mix it, and then they see the color, they see the taste, they determine the taste, whether it improves or not, the viscosity of the soup or the sauces. Okay, so what are the difference? Okay, what is the difference between us and them? Okay, the difference is in science. Okay, in science. Okay, so we are doing science, they are not doing science, they are doing art. Okay, so what is the difference between science and arts? Okay, in science, it is what is meant by science is a systematic and controlled, okay, with minimal bias and prejudice, okay, about something that we want to explore or you want to determine. Okay, so the key word is systematic, unbiased, and unprejudiced. Okay, whereas in arts, okay, okay, they don't have to comply with that requirement. Okay, so even our grandmothers, they put in something, they say that, oh, okay, it improves things. Those are experiments also, but it is not scientific. Okay, it is not scientific because there are so many aspects that are not controlled. Okay, so for example, if you were to put in compound A and it becomes more viscous, the soup becomes more viscous. So is it because of the compound A, or is it because of the higher temperature? Because they put turn on the flame more, so there will be more evaporation, then the soup becomes thicker or more viscous. Okay, we do not know. They also do not know because it is not controlled. Okay, so that's why in science, okay, everything has to be controlled. Then it becomes what we call as scientific. Okay, so in science, what we want to know is the truth. Okay, so why do we do experiments? Okay, determine the effects of variables, factors, okay, all of this. Okay, whether it is different or the same, any changes in the physical aspects, in the chemical aspects, and because we are in food science, okay, tastes, okay, aroma, okay, is very important. So from the sensorial aspect, okay. Sorry. Okay, so how do we do experiments? Okay, so the basic or the simplest approach, I think you also have studied this in your secondary and also primary school. Okay, sorry. Okay, so make constant and all variables. Everything has to be constant. Okay, except the factor we are interested in. Okay, so the term in that they use is pemboleh ubah yang dimalarkan. So make constant everything else. And then you have pemboleh ubah yang dimanipulasi. Okay, so so whatever that you make constant, okay, so that is not your interest, okay, because that's why we have to make it constant. Okay, so accept the factor that we are interested in. Okay, so if you were to look at this one that we discussed just now, okay, so for this example, everything we have made constant. The variety of the bananas, the weight of the bananas, the degree of maturity, because it is not our interest. We do not want to determine the effect of different varieties. It's not our interest. Doesn't mean that you can't do it. Okay, if that is your interest, then that should be your factor. Okay, we do not want to see the effect of the different weights. We do not want to see we are not interested in determining the effect of different degree of maturity. Okay, so that's why we make it constant. What we are interested in is to see the effect of temperature. So for the temperature, we will vary. Okay, we will vary, put it at different levels, vary it. Okay, so that we will be able to determine its effect. Okay. Okay, so whatever we we manipulate, so that is the factor. Okay, so that is the factor. Okay, so everything else is constant except the factor. Okay, the factor depends on the objective, because for this, the objective is to determine the effect of temperature, then the temperature is the factor. If it is to determine the effect of different variety of bananas, then the different variety of banana should be the factor, and the temperature should be constant. Okay. Okay, so when we do this, so any differences in the results, then we can say that it is due to the temperature, because everything else is constant except the temperature. Okay, but if we do not make everything else constant, for example, the variety is different, we are using different varieties of banana, then when we get the results, okay, we cannot attribute the results to the effect of temperature, because maybe it is due to the effect of different variety. Okay, so that is why when we do experiments, everything else all has to be constant except the factor. Okay, except the factor. Okay. Okay. So okay, so make everything constant except the factor. Okay, so the factor is what we are interested in. Okay, so any change is due to the factor. Okay. Okay, this is just an example. We have done that. So what is experimental design? That is the way and procedure that we conduct experiments or apply treatments in the lab. Okay, not on paper. Okay, because many, especially all of you will be presenting your proposals. Okay, research proposals. Okay, usually students will just put in any type of experimental design. Okay, so that is not correct. Okay, because experimental design is in the lab, not on paper. It is not being determined on the paper. Okay, it is being determined in the lab. So before you determine which DOE or design of experiment that you are going to use, you need to go to the lab. where you are going to do your experiment. Okay, where, and in more specifically, where you are going to apply your treatment onto your sample. Okay, so you need to understand what you are going to do, determine your setup. What are the equipments that you're going to do? What are the apparatuses that you're going to use? Okay, and preferably, okay, go in the lab and try it out. Okay, it doesn't mean that you have to analyze your samples, okay, but you need to try to apply the treatments onto your samples. Then you can get a better feel of what you need to do. Based on that, then you determine the design of experiment. Okay, for example, sorry. Okay, for example, okay, so for example, for this one, okay, in this case, if, okay, let me take another example, okay. Okay, so for example, if you want to determine, similar to the first one, objective to determine the effects of temperature and time of blanching, okay, on the color, etc., similar to the previous one, okay. But now you have how many factors? So what effects you want to determine? Temperature and time. So now you have two factors, right? Temperature, one, time, second. Okay. Okay. So in this case, for example, the levels, okay, for temperature, 60, 70, 80 degrees C. And for time, maybe it is 1, 2, 3. Oh, this is too much. Blanching. Okay, sorry. Okay, blanching, maybe you have like 10, 20, 30, 10, 15, 20 minutes. Okay, for blanching. Okay, so what are your treatments? Okay, so the levels, okay, this is a two-factor, more than one factor experiment. Okay, so the treatments will be, for example, you are going to do a factorial design. So you have your temperature, 60, 70, 80, right? And then for each, you will have your time, variation of time. Okay. Right. Okay. So meaning that your treatment is this one. The first, okay, treatment first is that one. And then this one. And then this one. Right. Okay. And then you also have this one. You also have this one. You also have this one. Then you have this one, and also this one. Meaning that for more than one factor experiment, okay, the treatment is a combination of levels. So treatment 1, 60 degrees C, 10 minutes. Okay. Number 2, 60 degrees C, 15 minutes. Number 3, 60 degrees C, 20 minutes. Okay. Number 4, 70, 5, 6, similar, and 8, sorry, 8, okay. 80 degrees C, 10 minutes, 10 minutes, and then 9, 10. Sorry, 7, 7, 8, 9. Okay, so those are your treatments. Okay, so you have to apply this combination. Okay, so but when you you go into the lab, you see that, oh, you only have one water bath. you only have one water bath. Okay, so if you do a factorial design, for example, okay, and you do randomly, okay, and you do it at random. Okay, for example, you do this first, and then sorry, this one second, this one third. Okay, for example, completely randomized factorial design. Okay, so first is 60 degrees C. Okay, or sorry, for a better example, like this is number 3. Okay, so first, what you need to do is you have to turn it on, increase it to 60 degrees C, right? Okay, and then for 50 minutes. And then second is 70 degrees C. Then you need to reduce it to 70 degrees C. When it has reached 70 degrees C, then only you can apply your treatment. Okay, put in your banana for blanching. But then after that, okay, you need to heat from 60, heat it up to 70, and then you need to cool it down back to 60 degrees C. Okay, so that is tedious. Okay, so as opposed to doing that, as opposed to using a completely randomized factorial design, might as well you use a split plot design. Okay, so we will be discussing this, but split plot means that you do one factor at a time. Okay, for example, you randomize and then you choose, okay, which temperature. So the temperature will be the whole plot, and then the time will be the split plot. So, for example, you randomly choose, so you do 70 first. So you do 70, set it at 70, and then do the different time. For example, 20 minutes, 20 minutes, and then 10 minutes, and then 50 minutes. Then only you change it to 80. Okay, so now it becomes more convenient as opposed to having to change the temperature every time. Now you only have to change it every 3 times of experiment. Okay, so it becomes convenient. So as opposed to choosing a completely randomized factorial design, better to use a split plot design, for example. How do you know this? How do you make this kind of decisions? By going into the lab and okay. Now it's back again. Okay. The line is not stable yet. Okay, because they are upgrading the line in UKM. Okay, so what did I say just now? Okay, so how do you make this kind of decisions? Okay, by going into the lab, okay, and then trying out your treatments, okay, and looking at the apparatus, the equipments that are available, what are the limitations. Then based on that, you decide on your design of experiment. Okay, so that is an example, okay, of what is meant that it has to be determined in the lab. It is not on paper. Okay, it is not just you look at the objectives. Okay, that is very important, but the objectives alone is not enough for you to determine a suitable design of experiment. Okay, so you need to consider the practical aspects and also need to consider the limitations that are present. Okay, so why do we need to use the correct experimental design? First and foremost, to eliminate bias, of course. Okay, because remember, we want to see the truth. If we were to do an experiment which is biased, then it is not going to yield us the truth. Okay, so whatever we get is not a representation of the actual happening, okay, in the population. Okay, and then also to reduce errors. Okay, errors will always be there, but we cannot eliminate errors, but what we can do is to reduce it. Okay, and as all of you know, errors are many, personal errors, and then you have experimental errors, and then you have other types of errors. Okay. Okay, but by having the correct experimental design, we will be able to minimize this. Okay. Also, different experimental designs, okay, will use different statistical equations. Okay, and that's why you need to understand the differences between the different experimental design. You should not be applying your treatment in the lab using or according to a completely randomized design. And then when you key in the data, the way that you analyze is according to a completely randomized block design. Okay, even though nobody will know, but actually you are not getting the truth. Okay, the correct answer or results. Okay, because different experimental designs is based on different sets of equations. Okay, for example, like if it is completely randomized block design, then the randomization is more limited. So when there is a more limited degree of randomization, then there is a higher degree of bias and error. But the statisticians, they have incorporate this within the equations that they use for calculating, okay, the sum of squares and everything. Okay, so that's why we need to use. So if we were to practically conduct our experiment according to a completely randomized design, and then we completely randomized block design, and then we analyze it using a completely randomized design, then that particular equation is not able to incorporate the limitation or the increased possibility of error and biases, okay, that we have carried out, okay, involved in when doing our experiment. Okay, so the result will not be the truth. Okay, to get the correct results, as we have mentioned just now, and also to arrive at the correct conclusions. Okay. Okay, so that is the introduction to design of experiment. So that's why it is very, very, very important for you to understand the design of experiment and choose the right one for your research experiments. Okay. Okay, so let's take a look at the first category of design of experiments. Okay, that is the design of experiment for one factor experiments. Okay, so now you know what are factors, okay, treatments and levels. Okay. It is very important to understand the basics. There is which one is the factor, the levels and the treatments. Okay, so that you can choose the right design of experiment. Okay, so let's take a look at the first type, the first one. This is a completely randomized design, or CRD. Okay, it is the most basic and the most simple. Okay, most basic and most simple, but not necessarily the most easiest. Okay, not necessarily the easiest one to conduct. Okay, basically, when it says completely randomized, so that shows that the randomization is complete. Okay, meaning that randomization will happen between all replications and all treatments. Okay, so it is completely randomized. Okay, so all application of treatments done without delay. Okay, and then there will no systematic order. All samples have the same probability to be selected. Okay, so example. Okay, let's take our example over here that we have done just now. Okay. Okay, so this is a one factor experiment. So we'll look at our one factor example. Okay, so that is we are going to determine the effect of temperature. Okay, the effect of temperature on color. Temperature of blanching, okay, on color in bananas. Okay, okay, so sorry. Okay, so what is our factor? So the factor depends on the objective. Okay, so that's why you need to really understand the objective. Everything starts with the objectives. Everything ends with the objectives. Okay. Okay. So the objective is to determine the effect of temperature. So the factor will be temperature. And how do we determine the effect? Then we have to vary the temperature. Okay. So we have the levels over here. Okay. So the levels. Okay, so we have decided that it is going to be 60, 70, and 80 degrees C. Okay, and then the treatments for a one-factor experiment, the levels are the treatments. Okay, the levels are the treatment. Okay, so the treatment will be 60, 70, and also 80 degree C. And the responses, okay, so in this case, we only choose for example, we choose one response, that is the brightness, degree of brightness, okay, under the CIE color system, that is the L star, okay. Okay, how many replications? 3 replications, okay, so 3 replications. So this is one factor experiment, okay, this is a one factor experiment. Okay, so how many experiments do we need to do? Okay, how many experiments? Number of experiments is, okay, we have 3 treatments, and then we are going to do 3 reps. Okay, so the number of experiments is 9. Okay, so the number of experiments is 9, meaning that the number of samples that you have to prepare, okay, is 9. Okay, so that is what we have to do. Okay, so what are the treatments? Okay, what are the samples? Okay, so I'll continue over here. Okay, so I'll continue over here. Okay, so we have our treatments. 60 degrees C, 70 degrees C, and 80 degrees C. Okay, and then we have reps. We have 3. So meaning when we prepare our samples, then sample 1 will be 60 degrees C. Okay, I'll just put 60. Okay, oh, sorry, I'll just put 60. Okay, 60. Oh. Okay, so it is 60 rep 1. Okay, and then also you will have 60 rep 2, and then also you will have 60 rep 3. Okay, 4, you will have 70 rep 1, 70 rep 2, 70 rep 3. Okay, and then you also have 80 rep 1, 80 rep 2, 80 rep 3. Okay, so these are our samples that we have to prepare, and then each of these we will subject to the different temperature. Okay, so 60 rep 1 we will blanch it in 60 degrees C. 70 rep 1 we will blanch it in 70 degrees C. 80 rep 1 we will blanch it in 80 degrees C. Okay, so these are our samples, okay, that we have to that we are going to prepare, and then we will apply the treatment. Okay, when we if we want to do this according to a completely randomized design, okay, completely randomized design. So completely randomized meaning that the randomization is between is between all samples, regardless of the treatment and regardless of the replication. So there will be a full randomization, a complete randomization between all samples. Okay, so there will be randomization between the different treatments and also the different replications. Okay, so when we have done this, when you enter your lab to prepare to do this experiment according to completely randomized design, you have to prepare 9 samples straight up. Okay, you have to prepare 9 samples. Okay, and then you have to apply the treatments. Okay, in a random manner. You do not want to have no systematic sequence. Okay, so you should not go into the lab and do this one first, and then after that you do this one, after that you do this one. Okay, so that is systematic, so we don't want that. Okay, or you go into the lab, you do this one, and then this one, and then this one. Also not correct. Okay, so it has to be completely randomized. Okay, so meaning that all of these have all these 9 samples have the same probability to be done first, have the same probability to be done second, have the same probability to be done third, and so on and so forth. Okay, okay, so what do we do? Okay, you can do it based using a computer to randomize it for you, because all computers have what they call as a random number generator. Okay, it generates random numbers. Okay, actually it is not totally random, because as we all know, computers, okay, they have algorithms. So meaning that when there is an algorithm, it is going to be systematic. Okay, so even though it is a random number generator, it is not 100% random, but it is good enough. Okay, so or you want to do it the simpler way is just to cabut undi. Okay, pick straws. Okay, so meaning that you write on a piece of paper, just write on a piece of paper. Okay, 60, and then this another piece of paper, this is another piece of paper, and so on. Okay, so you will have 9 pieces of paper, and then you fold it or you roll it up, put it in a container, and then mix it. Okay, and then just pick one. Okay, so when you pick one, okay, for example, you pick one. Okay, what comes up is, for example, 80 rep 2. Pick another one. So 60 rep 1. You pick another one. For example, it is 60 rep 3. And then you pick another one, 4. It is 70 rep 3. And then you pick another one. It is 80 rep 1. 6, 60 rep 2, for example. Okay. And then 7. Okay. What else? 70 rep 1. 8 will be mana yang belum? 80 rep 3. Okay. 80 rep 3. Oops. And then 70 rep 2. Okay. Okay. So this is cabut undi, picking straws. Okay. And you have this sequence. This sequence is what we call as a run order. Okay. This is our randomization. Okay. So when you go into the lab, okay, you prepare 9 samples. Okay, you prepare 9 samples. Okay, and then the first sample, do for this one, 80 rep 2. Okay, and then the second sample, do for 60 rep 1. Okay, so meaning that you have 3 replications. Okay, 3 batches of bananas. Okay, and then for each batch of banana, you have to peel it. Okay, and then cut it. Okay, for example, to the weights and dimension that you have determined. Okay, so for each replication, for each batch of banana, okay, you have 3 samples. Second batch of banana, you prepare 3 samples. The third batch of banana, you prepare 3 samples. Okay, so when you want to do 80 rep 2, so meaning you take 1 sample of banana from the second batch of banana, okay, and put it in the water bath for at 80 degrees C. Okay, so that is the first one. Then the second one, you take 1 sample of banana from the first batch 1, okay, and then you blanch it for 60 degrees C. Okay, and then for the third run order, third sample, you go and take 1 sample, okay, from the third batch number 3, and then you blanch it for at 60 degrees C. Okay, and for the fourth one, you take 1 sample from batch number 3, okay, and then you blanch it at 70 degrees C, and so on. Okay, so that is what we call as a completely randomized design, CRD. Okay, so you have to have run order for randomization. If not, there will be biases. Okay, but because it is a completely randomized design, okay, so the randomization is between the treatments, this one, between the treatments and also the replication. So you can see that the treatments are randomized, 80, 60, 60, 70, 50. Okay, it is not systematic. And also the replication also is randomized. Rep, second rep, first rep, rep 1, rep 2, rep 3, and then rep 3 again, back to rep 1. So it is randomized. So that is why it is called a completely randomized design, because the randomization is in total, okay, between all samples of the in that study. Okay, the treatment and also the replication. So that is why it is called a completely randomized design. Okay, so that is completely randomized design. Okay. Okay, so this is an example. Okay. So this is just a representation of what you can do just to keep to record your run order. Doesn't mean that you have to use this table. As long as you know run order 1 is which sample, run order 2 which sample, then that should be okay. Okay, which treatment, okay, and sample, so that should be okay. Okay, any questions? Okay, so after application of treatment, okay, we measure the parameters. Okay, so the measurement of parameters can be done directly after each run order or simultaneously for all samples. Okay, experimental design is regarding the application of treatment, not the measurement of parameters. Okay, what that means is that, okay, so like in our example here, okay, you have done the first one 80 for rep 2, second 60 for rep 1, okay, and then 7th 70 for rep 1, 80 for rep 3, 9 for 70 for rep 2. Okay, what you can do is that you can, for example, we start from the top. Okay, so the first, when you go into the lab, you prepare everything, and then you do 80 for rep 2. Okay, you do 80 for rep 2. When you have blanched this, okay, because the period of blanching is fixed. For example, 10 minutes. Okay, so for 80, okay, for this sample, this sample is from the second batch 2, and then We blanch it for 10 minutes, okay, at 80 degrees C. After that, after blanching, you can straight away measure the degree of brightness, okay? And then you do the second one. After blanching, after 10 minutes of blanching at 60 degrees C, then you can measure the degree of brightness. Similarly, after blanching at 60 degrees C for 10 minutes, you can measure the brightness. This would be ideal, but sometimes it is not practical. Okay, sometimes because of certain limitation, you can also not measure it, this one, but you just store it first. Okay, after this, store, after this, store, and then after all has been completed, then you measure. Also okay, as long as you have enough justification, okay, to say that during storage, there are no changes that will affect the result. Okay, so there are no changes that will affect the results. Okay, so how do you justify that? From the literature review, okay, from the literature, okay, previous studies that have shown that, okay, if for this sample, if you were to store it in this manner, in this condition, then it will not have an effect on the outcome of the analysis. Okay, okay, so that is what is meant by this statement. Experimental design is regarding the application of treatment and not the measurement of parameters. Okay, not the measurement of parameters. Okay, so it is up to you whether you want to directly, immediately measure, okay, after application of treatment, or you want to collect everything together, then only measure the response. Okay. Okay, so that is completely randomized design. Okay, another related, okay, related design of experiment is completely randomized block design. Okay, completely randomized block design. Okay, for example, okay, if in this case you are only using 9 samples. Okay, so what if, okay, you have temperature, sorry, you have the factor is temperature, but the levels, okay, so you want a wider range, so you have 60, 70, 80, 90, 100 degrees C. Right, so you have 1, 2, 3, 4, 5. Okay, and then your treatments, this is a one factor experiment, so the levels are the treatments. Okay, okay, and then sorry, your reps, for example, okay, for example, you want, it is you expect it to have a higher degree of heterogeneity, so you want to do 5 reps, for example. Okay, so number of experiment, how many? So you have 1, 2, 3, 4, 5 treatments. Okay, multiplied by 5 replications equals to 25 experiments. Okay, so it is 25 experiments. Okay, now okay, so what, okay, if you are going to do this according to CRD, so meaning that when you go into the lab, okay, you need to have 5 batch. Batch 1, batch 2, batch 3, batch 4, and then batch 5 of bananas. Okay, and then for each, you have to prepare 5 samples. So in total, you have 25. Okay, and you feel that this is too difficult because you have to handle 25 samples. Okay, especially when, for example, so meaning when you want to randomize it, you have 1, okay, 60 rep 1, and then 60 rep 2, 60 rep 3, 60 rep 4, 60 rep 5, and this one is 70 rep 1, and then over here you have 5, 6, sini 11. Okay. Okay, this is 80. And then this is 90. And then this is 100. Okay. Okay. So you have 25 samples. And this is very is difficult to handle. And also, you are concerned that when you do this first, for example, if the run order says that you do this, okay, by the time you do the last one, then something will affect it. It loses moisture, for example. So meaning that the samples have been affected. So now you are introducing a confounding factor. Okay. So because it takes too long a time until we get to this sample, for example. Okay. Okay. So if you feel it is less practical, less suitable, then another option is what we call as a completely randomized block design, or we call it as CRBD. Okay, so what happens is that for this one, we want to limit the number of samples that we handle. Okay, so that it is more doable. Okay, and because if there are too many samples for us to handle, not only that it will consume time, but also we will be more prone to make mistakes. So that will inadvertently increase the error. Okay, increase the error. So we do not want that. Okay, so another option is for you to do a completely randomized block design. Okay, and what happens is that okay, the principle is that if there are any confounding factors, or if there are any variation, okay, that is not the factor, other variations than the factor, then we block it. Okay, then we block it so that within the same block, everything will be uniform. So that is the approach. Okay, that is the approach of using blocks. Okay, and because for us in food science, we are most of the time we are doing experiments in the lab. Okay, in the lab it is more controlled. Okay, so it is more convenient in most of the time. It doesn't necessarily have to be, but in most of the time it is more convenient to take the replication as block. So we block the replication. Okay, so it is more convenient. Okay, for example, let's take our example just now. Okay, we have the factor is temperature level. Okay, we have just now 60, 70, 80, 90, 100. Okay, so that is the level. This is also the treatment. Okay, 60, 70, 80, 90, 100. Okay, replication, we have 5. Okay, okay, so what we if you were to do a complete randomized block design, okay, so what we need to do is to buy a batch of bananas. Okay, so batch 1. Okay, and so this batch 1, okay, we will divide it into 5 groups, where this will be, we will subject to 60 degrees C, another is 70 degrees C, 80 degrees C, 90 degrees C, and 100 degrees C. Okay, okay, and then we randomize it. Okay, so for this particular batch, okay, we have 60 rep 1, number 2, 70 rep 1, okay, and then number 3, 80 rep 1, 4, 90 rep 1, and then 5th 100 rep 1. Okay, so these are the sample because they are from batch 1 only. Okay, and then you determine the run order. Okay, so randomize all these 5. Okay, write it on a pieces of paper, mix them up, fold them, mix them up, and then pull pick straws. Okay, for example, number 1, run order 1. You pick 1, you get 100 rep 1. And then number 2, okay, you pick 1, you get 60 rep 1. Number 3, 90 rep 1. Number 4, 80 rep 1. And lastly is 70 rep 1. Okay, so this is your run order. So when you go into the lab, okay, you have 1 batch of bananas, you prepare them, okay, prepare the sample, weigh it, peel, weigh, and everything, and then you divide it into 5 groups. Okay, so the first, okay, so take the first group and then subject it to this treatment. The second one, subject to this treatment. The third one, subject to this treatment. The fourth one to this treatment. And lastly is the 5th one to this treatment. And then, okay, you can immediately measure L star, or you can combine everything, then measure. Okay, okay, combine and then measure. Also can. You can also do it like that. Okay, so now you see that, okay, there are randomization between treatments, right? But there are no randomization between replication. All the replication are rep 1. So there are no randomization between replication because we have designated the replication as blocks. So within the same block, all are rep 1, but the treatments are randomized. Okay, and then after that, okay, you take whether it is the same week or the same month or different weeks or different months, doesn't matter. Okay, you take another the second batch of bananas and then you repeat. Okay, you repeat this, but with different run order. You have to pick straws again. Okay, you have to pick straws again. Okay, so you will get, for example, number 1, it is maybe 60 rep 2. Number 2 maybe 80 rep 2. Number 3, 100 rep 2. Sorry. And then number 4, maybe 70 rep 2. And then number 5 is 90 rep 2. So you can see that, okay, between these two, okay, the run order is different. Okay, the run order is different. Okay, so that's what you want. So, and then and then you can measure immediately, or you collect everything and then only measure. Okay, so now you have finished your second replication, and you have to keep on until you finish your fifth replication. Okay, so then you average the data and you can run the statistical tests. Okay, so again, you can see that there is randomization between treatments, but there are no randomization between replications. That is why it is called a complete randomized block design, whether it is completely randomized, but only within the blocks. Okay, but only within the blocks. And you can see that it is more convenient, because as opposed to like this one, where you have to handle 25 samples all at once, but over here you only handle 5 samples. Batch 1 only 5 samples, and then batch 2 only 5 samples. Okay, so it is more convenient. Okay, so that is why it is called a completely randomized block design. Okay, okay, any questions? Okay, so we use when limitations or confounding factors exist that limit complete randomization. Okay, so different batch, different days, etc. So we need to create blocks, okay, to nullify the limitations or confounding factors. Okay, so within the blocks, it has to be uniform. Everything has to be uniform. Okay, randomization is done within the blocks only. Okay, within the blocks. Okay, so not all replications and treatments are randomized together. So there will be a limited systematic order. Okay, all samples within the blocks have the same probability to be selected. Okay, okay, so this is an example. Okay, so this is one example of how to record your run order. So if you can see that the replication, they are not randomized. So the first rep 1, and then followed by rep 2, and then followed by rep 3. Okay, so in a completely randomized block design, okay, there is a limited degree of randomization. And however, even though the randomization is limited, but it has been taken into account. Okay, has been taken into account within the models or equations that is prepared by statisticians. Okay, so that is why if you were to do a completely randomized design in the lab, and then you take that data and then you analyze it using a completely randomized block design, okay, as a completely randomized block design, then it will not yield the truth, it will not be totally correct. Okay, so that is why you need to understand how you do your experiments in the lab, and that it has also to be that also based on that also you will analyze your data using the software. Okay. Okay, so it is more convenient in most cases. I would recommend that students use the block design as opposed to a completely randomized design. Okay. Okay, any questions?
Doktor,
Yeah. Yes.
randomization ni kan kita kena buat in the same day jugalah kan, betul tak, the experiment?
Well, it depends on your sample. All the experiments, okay, can be done within the same day or on different days, as long as you have justification, because science is based on proof. Okay, that's why it is science. In art, you don't have to show proof. Okay, but in science you have to show proof. If you say that, okay, you prepare today, but tomorrow the samples will still be the same prior to being treated, okay, subjected to treatment, then it is okay. But in food, as we know that most of the time it is not okay because our samples are always changing, right? So that is why, but if you were to do a completely randomized design, it is more preferable for you to complete all the treatments within the same day in food science. Okay, because our samples are very sensitive and it always changes. Okay, and that will be quite difficult. So that's why in my personal experience is that I would prefer to use a block design as opposed to a completely randomized design, because you need to handle only a fraction of the number of samples at one time. Okay.
So doktor, kalau macam apa ni, the block design tu kita boleh buatlah macam replication 1 hari ni, esok replication 2 and lusa replication 3 macam tu kan?
Yes. And for example, like for bananas, I would really prefer to do a block design, because then you can take one batch in January, for example, okay, and then another batch in and then you finish that one batch, meaning that one replication, and then you analyze it, everything, okay, and then you go and get another batch, and then you do that also, so that the samples are representative of the bananas throughout the year. And it doesn't matter, because we are taking batches from different months, okay, or time period along throughout the year, okay, because we have blocked it. So within the block, it has to be uniform, not necessarily between the blocks, okay. And by doing so, we will be able to capture the variation that happens throughout the year within the samples.
Okay, thank you, Doctor.
Okay. Okay, any other questions? Okay, let me check. Okay. Any other questions? Okay. So if there are no questions, okay. I want to show you another what you need to do for practice 2. Okay, for practice 2, and this you will do. This one you will do. Okay. During your mid-sem break, and also when you on week 9. Okay, so meaning that, okay, next week is your mid-semester break. Okay, and then the following week, okay, there will be no lecture. Okay, so you can use that to do your practice 2. Okay, to do your practice 2, I just want to demonstrate what you need to do for practice 2. Okay, let me turn it on. Okay. Okay. So let me share with you. Okay. So this is practice number 2. That is for randomization. Okay. The objective of this is to demonstrate to you the importance of doing randomization and the effect of if not doing randomization, if there is confounding factors. Okay. Okay. So basically, you have this is a make believe. It is not real. Okay. I invented this. Okay. So this is just as a demonstration, for demonstration purposes. It is not real. Okay. So for example, you are going to do a study. Okay. A study, an experiment that involves 3 treatments. Okay. Involves 3 treatments. That is treatment A, treatment B, and also treatment C. Okay. And the results, okay, that each of this treatment will produce is for treatment A, the result will be 1. For treatment B, the result will be 5. For treatment C, the result will be 3. Okay. And because this is just a demonstration, okay, regardless of the replication, whenever you do a treatment A, you will still get 1. B, you will still get 5. C, you will still get 3. So meaning that if it is replication 2, you will still get 1, 5, and 3 over here. Okay. Okay. And then this one is the run order. Run order 1, 2, and 3. Okay. Okay. This is just a make believe. Okay. It doesn't happen in real life. But okay, what I created over here is that there is a confounding factor that will affect the results. Okay, according to the run order. Okay, meaning that okay, if the run order for the first, if the run order is 1, okay, there is no effect. Okay, meaning that if this is A, if the run order is 1, it is supposed to produce a result of 1. Then because it is run order 1, so there is no change. So the actual result will still be 1. Okay. Okay, but if the run order is 2, then the result will be changed by adding 2. And if the run order is 3, the result will change by a value of 4. Okay, meaning that, okay, for B, it is supposed to give a result of 5. But because it is run order 2, then the result will increase by 2, meaning that what we will see is sorry, is 7. Okay. Okay, and then for C, the measurement is supposed to, this treatment is supposed to give us a result of 3. But because it is done on run order number 3, so it will be added by 4, meaning that this also will give a result of 7. Okay. Okay, so so do this. Okay, first do it for sorry, no confounding factor, meaning when no confounding factor, meaning that there is no addition. Everything will be 0, 0, 0. This means no confounding factors. Okay. Okay, so no confounding factors. and no randomization. And then no confounding factor, but with randomization. Okay, and then with confounding factor, but no randomization, with confounding factor, with randomization. Okay, randomization meaning that, okay, with this one, if there is no randomization, the run order will always be 1, 2, and 3. So this is no randomization. Oops. Oh, okay. 1, 2, and 3. So meaning that there is no randomization. Okay, but if there is randomization, then you randomize this, okay, write on a piece of paper, okay, and pick straws, okay, cabut undi. So maybe, for example, you get 3 first, and then 1, and then 2, and then for this one, maybe you get like 1, and then 3, and then 2, for example. Okay, so this is randomization. Okay, so if there is confounding factor, if there is a confounding factor and randomization, so it will be like this: 1, 2, 3, 3, 1, 2, 1, 3, 2. That is randomization. And if there is confounding factor, then you will add 0 because it is 1, then add 0. Because it is 2, you add 2. Because it is 3, you add 4. So the result will be, okay, this is 153. Okay, so the result will be for this one, 1 plus 4 equals to 5. 5 plus 0 equals to 5. 3 plus 2 equals to 5. Okay, for this one, 3. Okay, because it is first run order, no change. Second run order, add 2. Third run order, add 4. Okay, so the result will be 1 plus 0 equals to 1. 5 plus 4 equals to 9. 3 plus 2 equals to 5. So that will be the result. This is for first rep, second rep, third rep, and then you average it out. Okay. Okay, so this is an example for with randomization with confounding factor. Okay, so if it is no randomization, then it will always be 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, and if it is no confounding factors, so it will always be 0, 0, 0, 0, 0, 0, 0, 0, 0. Okay. Okay, any questions? Okay, so I will post it, upload this in UKM Folio. Okay, and okay, and then so no lecture after mid-sem break. Okay, and then afterwards the following week, so you do your practice 2, and the following week we will discuss about more than one factor design of experiments. Okay, any questions?
Doktor,
Yes.
kita akan ada midterm exam tak?
Yes, ada. Midterm exam.
Minggu ke berapa, Doktor ya?
Okay, midterm exam lewat sikit. Biasanya saya buat lewat sikit. A little bit late. It will be as of now, it will be on the 7th of January.
Okay.
Okay. Okay, or better still. Okay, yeah, 7th of January. Okay, hold on. If we can do that. Okay, so let's I will give you the practice today, okay, and then you can do it during your mid-sem break, and then when we the week after that we'll discuss about more than one factor experiments, so that we will have more time to discuss later. Okay? Okay. So I think we'll do that. Okay, so I will upload the practice 2, and the deadline will be for submission will be 9th of December. Okay, so on the 10th of December, we'll discuss it and we'll continue with sorry. Okay. Okay, we'll continue with our lecture. Okay. Okay. Any other questions? Okay. So if there are no questions, okay. So we'll stop here. Okay. Thank you.
Thank you, Prof.
Thank you, Prof.
Thank you, Prof.
Thank you, Prof. Yusuf.
Thank you, Prof.
Thank you, Prof.